Welcome to today's webinar on the new G4 Next Generation Sequencing Instrument hosted by University of Minnesota Genomics Center and Singular Genomics. My name is Dinesha Wallach, and I'm the Managing Director for the UMGC, and I'm helping to facilitate this webinar today. Also with me today is Dr. John Gabi, our Bioinformatics Lead, Jerry Daniel, CLIA Manager and Genotyping Operations Manager, and Elise Froling, who is our NGS Operations Manager. On hand is Shay Anderson, Communications and Outreach Lead, and thank you, Shay, for arranging this webinar today for us. So for a long period of time, next generation sequencing has been dominated by Illumina. And due to the rapid advancement of NGS technology, there are new contenders now in the marketplace like Singular Genomics, Element Bio, and Ultima. As a core, we strive to keep pace and acquire state-of-the-art instrumentation, and we will be hands-on in assessment of this new and upcoming sequencing technology and how they can most benefit University of Minnesota researchers. We will look very closely at throughput and efficiency, data quality, range and applications, new data analysis methods, and of course, bottom line, cost. So as always, we value and invite PIs and researchers, their input to help us better understand your sequencing needs so that we can bring what's best suited for University of Minnesota core. Today, we will begin our exploration with Dr. Jordan Williams from Singular Genomics presentation on the innovative G4 instrument. Jordan, welcome, and thank you for being here.
Thank you guys so much for having me. I'm really excited to be here today with the University of Minnesota core. My name is Jordan. I lead our field applications team here at Singular Genomics. And I'll be talking to you today about our G4 sequencing platform. We also have Kyle on the line. He is going to be your dedicated FAS for Singular Genomics for the core over there at University of Minnesota. So you guys, a number of you may have already met him, but we're happy to be on the call today on this webinar. Feel free to ask any questions that you have, and I will get into it. So first, Singular Genomics was founded in 2016 to develop disruptive genomic solutions to accelerate the advancement of science and medicine. And 5 years later, we are now a publicly traded company based in San Diego, California. We have about 270 employees, and we've raised over $450 million to date. And about 300 million of that we still have in reserve. So we are extremely capital efficient, and we are in a strong financial position to support our many customers here to come. We have over 135,000 square feet of manufacturing, R&D, and office space. And we've issued we have 140 plus issued and pending patents protecting all of our technologies that we've brought forward. So as I mentioned, Drew and Eli founded the company back in 2016, and they have brought on a highly experienced leadership team of industry veterans. And we also have a very strong board of directors and a scientific advisory board full of genomics experts that we are excited to have. So today I'm going to be talking to you about our G4 sequencing platform, but we also have in development our PX system, which is our high throughput spatial and in situ cell analysis platform. This is something that is in the works and in development, but we are excited to bring it forward. We expect that it'll be something very disruptive in the field of spatial analysis. And if you're interested, please reach out to Deirdre, your Singular Genomics sales rep. So before we decided to create the G4, we talked to many NGS users to understand the primary contributors to kind of long turnaround times in NGS. And we learned that batching related delays and the time for sequencing were the primary contributors to long turnaround. So resolving those issues would require innovations to the sequencing chemistry, the fluidics, our optics, in order to shorten sequencing time, as well as allowing for higher flexibility in order to shorten the time needed for sample accumulation. And as you can see, with our many different R&D teams here at Singular, we've done that by shortening our sequencing time to around 2.5 minute cycle times. And because of our maximized flexibility with our 4 independent flow cells, we've shortened the time needed for sample accumulation because you can run what you have at a given moment. So with our G4 sequencing platform, we've targeted our 4 pillars to make the world's most powerful benchtop sequencer. We measure power as the data that is coming off of the system. This could be measured by gigabase per day, millions of reads per day, or we like to think of it as samples per year. And the G4 allows for between 15 and 400 gigabases of output per run, depending on the run size that you use. And you can see here that we have our 4 flow cells, and you can choose to load 1, 2, 3, or 4 of those flow cells at a time. And within each of those 4 flow cells, we have our 4 independent lanes. We also have industry-leading run times with 6 to 19-hour run times per run, again, depending on the number of cycles that you choose to run for any given sequencing run. And that means that for any of our sequencing runs, they will all complete in less than a day. And as mentioned before, the G4 has unparalleled operational efficiency due to it having our 4 independent flow cell positions, each with those 4 fluidically independent lanes. And this allows for independent ownership of those each of those 16 lanes. So no need to worry about sample index contamination, pooling of samples, maximizing a very large size flow cell. You have the freedom to kind of use 1 lane at a time or up to 16 lanes. Now, I say 1 lane at a time, meaning that one operator can use 1 lane while another operator uses the other 3. The minimum unit at a time for a sequencing run would be 1 flow cell at a time. And also with our novel 4-color rapid SBS chemistry, our accuracy is comparable to other sequencers on the market with Q30 scores ranging between 75 and 90% bases. So with our increased speed of less than 19-hour runtimes, you're not sacrificing your accuracy or your quality in any way. So here I'm showing a few graphs that highlight how those performance metrics stack up against other systems on the market. For power, we see on the x-axis the number of gigabases, and on the y-axis, we are comparing the G4 to 2 other systems in the market. The system I, or the Illumina system, is the NextSeq 2000, and system E, or the Element system, is the AVITI system. And you can see that the G4 is outputting as much data as a NextSeq 2000 and AVITI system combined. For speed, in the next column, we beat both of these systems by having less than 24-hour runtimes, while the other systems are around 48-hour runtimes for a 2x150 base pair sequencing run. As for flexibility, that can be measured in 3 ways. Either you have the number of flow cells, which is in this first column, you have your number of unique run sizes, or you have the number of lanes per run. And as you can see, we offer the maximum flexibility on the market by having up to 4 flow cells per run, 8 different run sizes owing to our 2 different flow cell size configurations, and up to 16 independent lanes per run. And as I mentioned before, as a researcher, it is important to have control over your samples. And with the option to own your own lane, that can be a game changer for how you design your experiments. No need to worry about index contamination or another library dominating your library on the flow cell. So with the G4 sequencing platform, we wanted to ensure that it fits seamlessly into the already established ecosystem. We've collaborated with around 15 leading library prep providers, showcasing the compatibility of our adapters with their workflow, with little to no impact on the upstream library prep. And I will get into that library prep and some options for conversion in the second half of this talk. Our system is easy to load with the user-friendly user interface that walks you step by step through the reagent loading process. And we also offer an off instrument management app, which will allow you to set up runs and monitor runs remotely. The G4 system outputs FASTQ files, which can be plug and play into any downstream bioinformatics pipeline that you're currently using. We use NVIDIA GPUs in order to accelerate our analysis. And we also work with a number of cloud providers to store your data off of the system. So we can hook up your system to that cloud or to your internal network so that data gets directly sent from the G4 up into your cloud or network storage solution. We do all of our base calling on board on the instrument, so no need for kind of any extra applications or clouds or anything like that. Everything happens on the G4 system. So getting into our offerings, we offer 2 flow cell types. The F2 is what's currently commercially available, and we will be launching with the F3 shortly. The F2 flow cell has a minimum output of 150 to 165 million reads. We'll offer 3 cycle configurations, the 100, 200, and 300 cycles. And those runtimes range from 8 to 19 hours. And with the quality scores, again, at 75 to 90% bases above Q30. For our F3 flow cell, we're going to offer 300 to 330 million reads for that flow cell. And we're going to tack on an additional cycle configuration of 50 cycles for that kit. And those runtimes will be between 6 to 8 hours. So all of our runtimes will be between 6 and 19 hours for our traditional flow cells F2 and F3. We'll also be providing a max reads kit, and this will allow for up to 1 billion reads per flow cell for short counting applications, and that will be 4 billion reads per run. And so I do want to emphasize that these are 150 million reads per flow cell, and we're allowing 4 flow cells per run. So you can multiply this number by 4 for the max output. So we have 8 different run sizes, our minimum being 150 million reads for one F2 flow cell. And if you were to load 4 F3 flow cells, you would get a max output on the system of 1.2 billion reads. We have a few white papers highlighting some key applications. I will discuss some of these later on in the presentation, but you can find these white papers and our third-party collaborator posters on our website. So briefly, to get into pricing for the instrument, the list price is $350,000. And we do offer volume-based discounting and some flexible financing, such as leasing and some other creative solutions. So please reach out to your Singular sales rep for more information there. And when looking at this price of the $350K, keep in mind that number, keep in mind that our system, the G4 sequencing platform, can output the same amount of output as a NextSeq 2000 and a NVIDIA combined. In terms of reagents, our flow cells range from $600 to $1,000, which for our lowest price flow cell will get you down to $150 per lane, which we're really excited to offer. So experimenters can purchase, 1 lane from a core and be able to press go with only $150, which we think is going to be a game changer. And as mentioned on the previous slide, with our max read kits, we're able to get down to $1 per million reads. And we're also able to achieve around $8 to $10 per gigabase, but we don't usually like to talk about our price in terms of gigabases because with other companies that do this, it isn't necessarily reflective of the price the customer pays unless they're maxing out the flow cell to its optimal efficiency. And we all know that oftentimes that isn't the case. You're either needing to move quickly, so you don't want to wait to pull samples on a large flow cell, or it's an odd size flow cell, so you just can't hit the max output. And that is shown in my next slide here. So here we are showing how the G4 can not only increase operational efficiency by increased run size options, but it also increases cost savings because there will be less unused space when using the G4 sequencing platform. So on the x-axis here, you can see the different flow cell configurations. So for the G4, we have the F2 here, and then one run with the F2, one run with the F3. Here would be a run with the F2 and F3 together. Here would be one with F2 and F3 together, two F3s together, and so on and so forth. Here is sequencer I, or this is the NextSeq 2000, and you can see that it has your 3 run options. And you can see with our mini run size configurations, you're able to find the one that best fits your experimental parameters without sacrificing on cost. And you're able to run the samples you have at any given moment without having to wait to pool. And that allows you to decrease your turnaround time and also increases your operational efficiency. So I'm going to get into kind of the second half of my talk here, which is more into the G4 workflow and how we can look at the library prep, our quality, some different data associated with our applications. So first, we're going to talk about library prep. So we have a couple of different library prep options. You can create a de novo singular library prep. We will be selling adapters for PCR and for PCR-free workflows. And we will also be selling indexed singular genomic primers with 96 unique dual indices. Those will be 12 base pairs long, but the user can choose to use between 8 and 12 of those base pairs when sequencing. We'll also be providing non-indexed singular PCR primers. You'll also be able to use those PCR primers for conversion of existing libraries. So we have 2 options for conversion. We have option A, which I'll get into both in more detail, which option A will allow for the user to keep the original index information. So that would be useful if your library is already pooled and you want to convert from an already pooled library to a singular library. And option B is nice if you don't mind replacing your previous indexing information with our singular index information. So this is best used for single libraries, maybe libraries that weren't indexed before. So let's first talk about de novo library prep. And this will look very similar to the library prep that you guys have seen maybe from NEB. So you would start with your DNA, you'd fragment it, and repair with your 3' overhang. You then ligate on your adapters, and notice how the SP1 and SP2 adapters here, these would be your traditional sequencing primers that you would use even without singular sequencing. These adapters would have an excisable base here, and you would use an enzyme to excise that base, creating these Y-shaped adapters here. Then you would just amplify with our singular adapters. Shown here are our indexed adapters, but you also have those non-indexed adapters. And those would elongate to add on our singular specific flow cell adapter sequences. In this case, for PCR1, it's S2. And then with your second PCR cycle, you would add on your S1. So our library structure looks very similar to what you're used to with sequencing by synthesis sequencing platforms. You have your DNA insert here. You have your sequencing primers here, SP1 and SP2, which would be synonymous with like an Illumina true sequencing primer. You have your index here, which for singular would be 12 base pairs. And then you have your singular specific adapters, which would be S1 and S2. So your library prep would look very similar to what you're doing now. The only difference would be maybe you grab a different tube of primers from your freezer. Instead of your traditional library primers that you would use, you would just grab the singular tube of index primers. Now, moving on to conversions, as I mentioned, we have 2 options. So for option A, you would be keeping your indexing information. And with this option, we would be annealing our primers down to the already integrated indexed, or excuse me, adapter sequence here. So we'd be annealing down to the already placed adapter and then elongating with our S1 and S2 singular specific adapter sequences. So from here, you may retain a little bit of that previous adapter sequence, but you're able to maintain the indexing that was there. while also including the S1 and S2 singular specific adapter sequences. Now, this can be done with a quick PCR amplification. So it'd be about 6 or 7 cycles of PCR. So this can be done with a quick conversion, maybe 30 minutes long in the lab. Now, the next conversion is a similar conversion where you're just using PCR primers again, 6 to 7 cycles of amplification, but this time we're laying down on those SP1 and SP2 sequencing primers, and then elongating with our indices and our sequencing, or excuse me, our singular specific adapter sequences here. So again, our conversion processes, they would be very quick, very efficient PCR reactions in order to get our singular specific S1 and S2 adapters onto your already made libraries. I'm happy to answer any more questions about that process after the webinar, or you can direct those questions to Kyle, your FAS, if you have any kind of specific, maybe a little one-off questions on how you can adapt your already made libraries or your current workflow to the singular specific workflow. So next, I'm going to quickly discuss our instrument loading and run kickoff. This is the G4 sequencing platform, and you can see that we have all of our doors open here to reveal our different reagents. At the top here, we have our sample loading cartridge, and that's where you would load your library. So each of those cartridges has 4 independent wells that aligns with the 4 independent lanes on the flow cell. We have our reagents here. Down in this bottom compartment, we have your hazardous waste disposal, as well as your 4 wash cartridges. And then over here, peeking behind, we have our flow cell compartment. Now notice that we have 4 compartments for the 4 different flow cells on the instrument. So you're only loading reagents for the flow cells that you choose to run. So if you only want to load one flow cell, you only load reagents for that one flow cell. You aren't wasting reagents if you choose to go with a smaller run size. So we are maximizing the flexibility of the system here. And although this picture is a little small, I meant to zoom in, you can see that this is our user interface. It's extremely user-friendly. It is a touch screen. You just kind of click sequence, you type in your parameters, and you click go, and the system will walk you step by step through the process of loading each of these reagents into the system. I'm not sure how many of you guys have had the pleasure of seeing one of our demos at one of the conferences, but it's pretty fantastic. It's really easy to load the system in about 15 or 20 minutes or less, and that's kind of with talking through a demo and answering questions. Usually can load very quickly. So now to get into more of the data coming off of the G4 sequencer, we're going to look at some quality and GC coverage. So for this slide, we're looking at whole genome data that was produced on our G4 sequencing platform from the human cell line HG002. This is a PCR-free library that was made, and we sequenced it at 2 by 150 base pairs. And you can see in column 1, we have read 1, and in column 2, we have read 2. For both of our reads, and you'll also notice that there's 4 lines here, maybe hard to see, but those 4 lines are associated with the 4 flow cells on the run. So you can see that with each of our 4 flow cells, we get very similar results getting output from the system. And for read 1, you can see that we have a very high percent GC, a very high percent Q30, and we have a 99.8% accuracy. And then for read 2, you can see very similar metrics. So we're not losing any quality or accuracy with our read 2. You can see similar for our GC content down below, that we have even GC coverage across the entire genome between 10 and 80% GC. So moving on, for the first column here, we see 3 bacterial genomes. We have the PHEP here, which has a lower GC content, around 42%. We have E. coli, which has a kind of run-of-the-mill 50% GC content here in black. And then we have our M. ruber, which has a higher GC content around 63%. And you can see that for each of those bacterial genomes, we're able to call the GC content accurately, and we have minimal GC bias across those 3 bacterial genomes. You can see that we maintain our accuracy of 99.8% across those 3 varying GC content bacterial genomes as well. On the right-hand side here, we see our human genome. Now this is with HG001, and just looking at normalized coverage for the GC content of targets across the genome. Again, very good coverage between 10 and 80% for that window for the human genome as well. I'll get into a little bit more detail with the human genome in our application-specific data to come. So next, we're going to look at our index validation and our lack of index hopping. So we ran 4 lanes each with 24 indexed Salmonella whole genome samples using different unique dual indices for each lane. The data were analyzed to detect any unexpected dual index pairs between the 24 dual indices, which would be indicative of index hopping events. And as you can see in the figure, index hopping events were extremely rare. So if we highlight, let's say the first box here, we can see we have indexes 1 through 24, and 1 through 24. Because they're unique dual indexes, index 1 is going to pair with an index 2 for that same primer pair. So we expect to see this kind of dark band across the middle where index 1 is appropriately paired with index 2. We shouldn't see any dark boxes outside of that line. And we have calculated that our index hopping is around 0.07%, so less than 0.1%, which is much less than other sequencers on the market. We are routinely demultiplexing at greater than 98% of the reads. And as I mentioned, we have 96 unique dual index primers, each of those containing 12 base pair barcodes. One feature that I'm really proud of on the G4 is that we handle low diversity libraries very well. And that can be seen here in a couple of different experiments. So first we sequenced a methyl-seq library, so an EM-seq library. And for these libraries, they're somewhat low diversity in that for the most part, 3 of the 4 nucleotides are overrepresented. And so some users would tend to use a more Phi-X in order to increase the diversity of that library. So here you can see that we sequenced that same library with either 0% Phi-X, 5% Phi-X, 25 or 50% Phi-X. And you can see that even with 50% Phi-X or even down to 0% Phi-X, we're seeing the same Q30 metrics. And in fact, across the board with all of our metrics for these libraries, they looked fairly identical. So that allows you guys to save more money and not having to run Phi-X in order to increase your diversity. So it's definitely a cost savings there that we handle low diversity libraries well. Another example here is from one of our external partners where they had their own proprietary library structure. And at at position 22, each library had an A tail. And you can see here for Illumina sequencing, they see a drop in quality down to, looks like maybe Q10. But for singular sequencing on the G4 platform, we maintained our high quality across that low complexity base and throughout the reads. All right. And lastly, let's get into some application-specific performance. So it was important for our system to be highly versatile and to address the variety of applications that our customers might see on a daily basis or even on a weekly basis. And it's important that you can scale throughout these with very minimal price difference at a cost per sample basis. So we want to highlight our flexibility and our cost efficiency. So first, we're going to look a little bit in depth at our RNA-seq data. And this is just a graph highlighting what a possible run could look like. So it would be maybe a 12 to 15-hour run. If we're doing a 2x100 base pair run, 50 million reads per sample. If we are doing 50 million reads per sample, we could do 3 samples per flow cell on an F2 flow cell, or 6 samples per flow cell on an F3 flow cell. If you were to run all 4 flow cells, that could be 24 samples per run. Now, in this experiment, we sought to assess the accuracy and technical reproducibility of the G4 platform. To do this, we prepared libraries from UHR and spiked it in with ERCC control RNA to examine accuracy over a wide dynamic range. And we sequenced on 2 F2 flow cells, which were split across 2 separate flow cells. So we have 2 technical replicates. It's important to note that we downsampled to 25 million reads per sample for this analysis. We sequenced at a 2x150, or excuse me, 2x100 with 12 base pair indices. Following sequencing, transcripts were quantified using STAR and Salmon. And of note, we achieved greater than 85% Q30 scores for both of our paired reads. So overall, you can see here for our 2 technical replicates, we observed a high correlation of ERCC counts in both replicates in the figure here on the left. So expected versus observed was highly correlated. And on the right here, you note the correlation in gene counts between Rep1 and Rep2. So this speaks highly of our technical reproducibility on the G4 platform. In this portion of the analysis, we compare the UHR libraries that were prepped from the same sample source, but prepared for both the Illumina NextSeq platform and the G4 platform using our primers. So correlation analysis here on the top left graph shows the similarities in RNA across the platform. So this is the G4 here compared to the Illumina platform, and you can see that we are highly correlated, where the G4 and the NextSeq 2000 show similar unique gene counts based on different transcripts per million bins. A higher transcript per million bin here versus a lower transcript per million. And so overall, you can see that the G4 and the Illumina platform perform very similarly. Furthermore, coverage uniformity seen here in the bottom left screen across the 5' to 3' positions using normalized coverage virtually overlaps with both platforms. So you can see the G4 and the Illumina instrument here are overlapped entirely. And then using STAR for RNA-seq analysis, the read distribution across different regions of the transcriptome are nearly identical across the platforms. This provides confidence that the G4 can serve as a suitable alternative sequencing platform to the Illumina platform. Next, we're going to get into our single-cell RNA-seq analysis that we did on site. For this study, we created Singular Genomics specific 10x Genomics single-cell 3' libraries. We used fresh frozen PBMCs for the RNA coming out of there. And we sequenced the libraries on the G4 across an F2 flow cell. For this experiment, we targeted 7,000 cells per sample with a sequencing depth of 20,000 reads per cell, which was based on 10x Genomics recommendations. But as you can see here, we achieved over 9,000 cells per sample, and we were able to hit our around 20,000 reads per cell with our sequencing run. Of note, our Q30 scores were well within our specifications of 75 to 90%. And the single cell metrics are on par with Illumina data, which I will show in more detail on the next slides. I do want you to see that for our 2 reps, we had about 180 million, 190 million reads output from our F2 flow cells. So that is over our minimum of 150 million reads that we guarantee. Okay, so we observed strong correlations of pseudobulk profiles not only between the technical replicates on the G4, as seen here, replicate 1 and replicate 2 with an R² of 0.99, but also between the 2 platforms. So here you can see the G4 on the y-axis and the NextSeq on the x-axis. Again, really high correlation scores with our R-squared at 0.98. We also performed UMAP dimensional reduction with the combined G4 and NextSeq datasets. We observed nearly identical embeddings across platforms with comparable representations of T cell, monocytes, and B cells as inferred by the gene marker expression. Furthermore, to perform a more in-depth and biologically relevant comparison, we performed unsupervised clustering and an automated cell type annotation with cell typist on the combined data set. And the adjusted RAND index, as seen here, which is a measure of similarity between sets of labels, demonstrated nearly identical cell type and unsupervised cluster labels between the G4 and the NextSeq datasets with comparable lineage marker expression profiles. You can also see down here below that the cell annotations across platforms were nearly identical as seen by the heat maps associated with the canonical PBMC lineage marker expression profile. So now to get into some exome data. Again, we're doing a 2x100 base pair run and with an estimated output of 35 megabases at 100x coverage across the genome. So for this experiment, this slide is the results of our training and testing of deep variant for exome sequencing. And on the left, you can see some key metrics from the validation set. So we had around 110x coverage of the HG001 genome. And we can see even coverage over a range of GC content, further illustrated by the fold 80 down below. If we take a look at the table on the left, there's the performance of the default deep variant model at version 1.3, and then our trained model here, the singular model. So you can see that there is some improved performance with our singular trained model for version 1.3. And for version 1.4 of the deep variant model with the default versus our singular trained model, we see even greater improvements with the metrics coming out of our singular trained model. And so this just shows that we have improved results now looking into our de novo model building. And we're really excited about this strong performance that we see. And lastly, to get into some, well, not lastly, we have two more after this, but to get into our target enrichment, we have some newer data coming off of our G4 sequencer. So we're going to move on to somatic variant detection, where we have been testing the Parabricks accelerated UMI error correction pipeline. So in this experiment, we start by pooling a set of 23 reference cell lines to create a control, compromising many variants at 2% or greater frequency. That material is used for library preparation via the IDT XGen cfDNA kit. And as noted on the right, this kit adds UMIs to complementary strands of DNA. And the UMIs are used for single read family error correction to yield error-corrected representatives of each starting DNA molecule. And as you can see here with the G4 compared to the NextSeq, we have very comparable and highly correlated data coming off of the sequencer. In this graph here, you can see the observed allele frequency versus the expected allele frequency for the G4, so highly correlated with what is expected. And then here you can see the G4 compared to the Illumina NextSeq 2000, again, very highly correlated across the platforms. That is even further highlighted down here with these Venn diagrams, with the G4 comparing truth in gray to the Illumina here in blue. Finally, we're going to look into some of our whole genome data. And we can see this is done on PCR-free human genome library HG002. We did 2x150 sequencing on the G4, and we were able to achieve around 31x target coverage. And this data is looking at the trained Google Deep Variant version 1.4 that was deployed through NVIDIA Parabricks. And you can see our results here, 31x coverage, our accuracy is stellar at 99.8, 99.9%. We have very low total duplication rate, around 4.1%, and even lower optical duplication rate, around 0.29%. Our SNP precision is awesome at 99.86%. SNP sensitivity is great at 99.1%. And that is maintained through our indel precision and indel sensitivity as well. Now, I think I have maybe 3 minutes left to chat, and I wanted to mention some of our specialized applications and some of our novel kits. So we have our max read kits here on the right, which I mentioned a number of times throughout our presentation within the flow cell. And for the sake of time, I won't get too deep into the weeds, but we are able to sequentially sequence multiple sequencing primer sets that have pollinated the same well on the flow cell. And this maximizes the output of the flow cell in that we can get 4 times the output of a flow cell previously. So we're able to get 1 billion reads per flow cell, meaning around 4 billion reads per run with our max read kit. And then with our HD-seq kit, we're able to pair both the top and bottom strand of the DNA duplex using a bobby pin adapter here. And then once we go into sequencing, we're able to sequence the bottom strand with our read 1 primer and the top strand with our read 2 primer. And that allows us to get rid of any strand-specific DNA errors, reducing the number of DNA errors that you see in your sequencing and highlighting only those real variants that are seen in both strands of the DNA duplex. So it's our version of consensus kind of duplex sequencing that we're excited to offer. To get into some of the quality scores seen with our max read outputs. So with that max read kit, we have the ability to do 8 sequential reads with that max read kit. And I just want to show here that with those 8 sequential reads, we are not losing anything in terms of our quality per bases. We're still maintaining a high Q30 of over 80% for each of our reads with those 8 sequential sequencing modules. So in this experiment, we looked at 8 50 cycle reads, resulting in over 250 million reads per lane, or 1 billion reads per flow cell. And you can see that we maintained our even gene coverage uniformity across the different, these are 4 different cell lines here. And we were able to have still very high correlation in gene expression comparatively against the NextSeq and the G4 with our max read output kits. So to summarize, I've shown you the benefits of the G4 system. It's fast, it's flexible sequencing, and it's accelerated variant detection without compromise in data quality. And the G4 fits into the existing lab ecosystem and enables flexibility by having the ability to own your own lane. And the G4 shows high concordance with the leading NGS platforms across many different applications. And with that, I'd like to say thank you so much, and I'm happy to take any questions that you guys may have. I see one in the chat. Is this system compatible with TotalSeq from BioLegend? I'm unfamiliar with that. Maybe you can elaborate. Is it a library prep? I can't hear anything. If you're talking, okay, I see. I'll wait for a little bit more clarification on the TotalSeq. I have another question that's come up. What is the time frame for shipment and setup for the G4 instrument? Are the supply chain issues announced during the summer still delaying your hardware manufacturing process? That's a great question. So right now, if you were to order a G4 system, the time for that to get installed would be about 3 months, 3 to 4 months. And then that installation and training would happen within a week's time. So you get your system installed and you get trained on that system within the same week, depending on your time frame. I fear that I may be missing some questions. I can't hear anything. I don't know if it's my system.
Jordan, no, the rest of the guests will be asking the questions through Q&A.
Okay.
So while we wait for another question, can custom primers be added to your sequencing?
Yes, great question. Sorry, I didn't mention that in the library prep setup, but you're absolutely able to use custom primers. Those custom primers would have to fit into certain parameters, and they would fit into those parameters that you would traditionally see on other platforms. But you're absolutely able to use custom sequencing primers for both of your inserts.
Just a quick follow-up on that, like, does it have its own place where you can add the custom primer? Yes, okay.
Yes, so you would add those custom primers in our reagent cartridge, and we have explicit instructions on how to do that as well.
Thank you.
So I have another question coming in. When converting libraries to be singular compatible, are there efficiency quality differences between the converted sequencing and the original sequencer data? That's a great question. And we don't see that. So with the few customers that we've had send in their converted libraries, they've compared it to their existing sequencer, and they don't see any noticeable differences in data between those. There's one caveat, and that's you will be adding a few extra amplification cycles, 6 to 7. So if your particular application is dependent on a low number of amplification cycles, you may want to think about doing a de novo library prep, or you can kind of have a little bit of wiggle room with your conversion and try to reduce the number of cycles that you do in order to reduce the number of amplification cycles. But it's a very highly efficient process. Oh, okay, great. We got some clarification. TotalSeq simultaneously detects surface proteins and RNA with our TotalSeq antibodies, panels, and hashtags, which integrate seamlessly into existing single-cell workflows. That's awesome. That's really cool to hear. I'd love to learn a little bit more about that, and maybe Kyle and you, Claire, can set up a conversation to discuss a little bit more about that application and how that can better fit onto the G4 sequencing platform. I have another question here. Is the G4 similar to MinION? I would say that it's not. So our system is 4-color chemistry sequencing by synthesis. We have our own proprietary novel application for sequencing by synthesis. So it's more similar to an Illumina sequencer, and that is seen by the data that comes off of our instrument being very similar to data that you would see with an Illumina sequencer. It's more similar to like a MiSeq, since we do utilize our own proprietary 4-color chemistry. Okay.
Oh, sorry.
No, go ahead.
Yeah, Jordan, I was wondering, do you have a method for assessing the library viability beforehand?
Do I have a method for?
Yeah, so I guess we have traditionally used, qPCR kits that come with P5, P7 primers built in that come packaged with the kit. Is there going to be something similar for this type of library?
Yes, we will have our own similar primer set that will be targeting our S1 and S2 adapters in order for you to do those qPCR assays. Yeah. One other, let me see, maybe I can stop sharing my screen so we can start seeing each other. And I had another question come in. For GeoMX spatial projects, we need 27 by 27 paired in. What would be the best option? What would be the option to flow cell and output in millions reads? Okay, so that would be a good fit for our 50 base pair kit with our F3 flow cell. And that F3 flow cell would output a minimum of around 300 million reads per flow cell. So you can get your 50 base pairs, so 27 by 27. We do include, and this is top secret. We include 50 extra cycles in each of our kits. So our 50 cycle kit would actually be 100 cycles. And then our 300 cycle kit's actually 350 cycles. So that will allow for you to kind of have a little bit of wiggle room in terms of the indices that you put in, as well as any kind of extra base pairs that you may want to tag on to some of your runs. And we do support runs that aren't, wow, my brain is blanking on me. But for instance, we allow like a 28 by 91 base pair sequencing run, so they don't have to be synonymous in terms of the reads per the cycles per read.
Are there limitations on, oh, sorry.
No, go ahead.
Are there limitations on the configuration of your cycling? So for indexing, so there's single cell and spatial runs that require an indexing read of maybe 28 cycles. Are there any limitations there as far as any configuration just with the additional 50 cycles?
Yeah, so you said that, so yeah, for that one, for that 28 base pair read, that can fit nicely into our index one read. For our insert one read, for our indices, we're gonna cap that at 24 base pairs for our index reads. And then the minimum will be 8 base pairs. And then for the actual insert, the max length that we will support is 150 base pairs, but we'll allow the user to go up to 200 base pairs, knowing that there'll probably be a drop in quality for those last 50. Did that answer your question? Okay.
What fragment length is possible to be sequenced? So the library insert length, what range of inserts are possible to be sequenced?
Yeah, it's going to be very similar to what is possible on the Illumina sequencer. So it's like the sweet zone is between 200 and 600 base pair insert sizes.
And is there any notable issues with very small library fragments? I think on, some of the Illumina sequencers are very sensitive to small fragments and will, if you have relatively small amounts of small library fragments, the data you get back will be primarily those small fragments. They'll kind of outcompete the larger ones. Is there a super strong bias or a moderate bias for length?
Yeah, we do. I wouldn't say that we have a super strong bias, but we definitely do have a bias towards shorter fragments just based on how we do our clustering. So our clustering does tend to bias shorter fragment lengths.
And is this a pattern flow cell? This is
It is.
It is a pattern. Okay.
Yeah.
And can the different flow cells on the instrument be started sequencing runs at different times, or do you so are they like fully independent, or do you start a batch of whatever flow cells you have loaded and they run, and when they're done, then you can swap out?
Yeah, that is a common question, and it is not, we do not have a random access instrument. So the moment you start the run, that run must complete before you load in your next round of flow cells. We do believe that since we have such fast turnaround times of less than a day, that hopefully wouldn't, impede your operational efficiency. You could just throw on those next 2 or 3 flow cells the next morning.
Mm-hmm. And are there any publicly available data sets, FASTQ files from your instruments?
Yes, there are. So you could, if you go on our website, I think you can ask for those, but you can also just email myself or Kyle, and we'll be able to hook you up with those data sets. Just trying to see if there's any other questions in the chat. Oh, wait, I see a couple. Do you have a method for assessing library? Oh, we already answered that one.
Yeah, sorry, that was my question.
No problem. I have a problem reading and talking at the same time.
In your remote monitoring runs, is that in real time?
Yeah, so that's definitely the goal. So when we're ready to launch that platform manager app, you will be able to monitor those runs in real time from your off instrument app.
And what operating systems are supported for that app?
Great question. So right now, the only operating system is the Windows platform. We are our future goal is to get that as web-based so that it is, agnostic towards the operating system. But right now, it's just Windows-based.
When you said the lanes are individually addressable, but within the flow cell, but if you started, a flow cell with only 2 lanes that are loaded, it's not like you reuse the other 2, right? No.
That's correct. So each lane is individually addressable, meaning that if you have the same barcode in each lane, they would get demultiplexed separately. So you don't have to have the whole rigmarole of making sure that your library doesn't have the same indexes as somebody else's library from a different lab. However, the smallest unit for the run would be the whole flow cell. So we do recommend using all 4 lanes of the flow cell at a time. But that's why we give the option of our F2 flow cell with that 150 million read output. So that's around 37 up to 50 million reads per lane for that flow cell. And then our F3 flow cell has 330 million reads, and that's closer to 75 million reads per lane. And so we try to make it as bite size of a piece as possible. So you have 4 bytes in that flow cell, but you have to kind of have at least 4 bytes in that meal.
You can do demultiplexing on the instrument. I assume you guys also have software that allows demultiplexing off the instrument.
Yes, we'll make our demultiplexing software available so that you can do that off instrument if you'd like.
And will that be like command line software that can be run on a variety of operating systems?
Yes, that's correct. Yeah. These are great questions. Please keep them coming.
Regarding the conversion of the libraries, so is there a cleanup step? What else is involved once you do the PCR? What do you recommend?
Yeah, that's great. We have a quick protocol, and it's just a quick 6-cycle PCR. You add in your primer pool and your enzyme, and we recommend around 2 nanograms of your previous library. You amplify it up, and then you do a bead purification. We recommend between 0.x and 1x volume ratio to your beads, depending on the size of your library, just to get rid of any extra primers that may be hanging around. And then you're ready to go. Denature, dilute, and put it on the system.
And it's not a kit that comes with yours to convert, or we can use any enzyme, any PCR that we choose, or is it a kit that you normally sell?
Yeah, we'll recommend a kit. We have it in the works to sell a conversion kit. We aren't right now. We have the primers available, but we can definitely send you some recommendations. We've done it with a number of enzymes internally, but we have our recommended ones. So Kyle and I can discuss that with you, what best fits your lab.
Well, it's getting close to 2. If there are no other questions.
Well, thank you, Shay, for putting Kyle and I's email into the chat. Feel free to reach out to either one of us if you guys have any other questions, or if you want any more information on the G4 sequencing platform. We're happy to answer them.
Thank you. That's great. So thank you everyone for your questions and for helping the UMGC access the capabilities of G4 instrument. We value your input, so contact us at next-gen@umn.edu to share with us your impressions and how the G4 would impact your research. And watch your inbox for spring webinars, because we will be hosting the other NGS contenders in the market. And with that, we are going to conclude our webinar. And again, thank you all for attending. And many thanks to you, Jordan and Singular Genomics, for your time and information presented on the G4.